IQAC Kumaon University Nainital, Dr. Dipendra Singh, Member DCI, Assistant Professor Pandit Ravi Shankar Shukla University Raipur, Professor B.S. Goop, UIPS Coordinator Punjab University Chandigarh, and Dr. Kumar Gaurav, Principal Guru Gobind Singh College of Pharmacy, Yamunanagar. I welcome you, sir. I also extend a heartfelt welcome to all the participants. For the first technical session, I would like to introduce Professor Kumud Upadhyay, Deputy Director, IQAC, Kumaon University, Nainital. He has more than 22 years of teaching and professional experience. He presently holds the post of State Coordinator, Indian Society of Pharmacognosy, and General Secretary, IPGA Uttarakhand, editor of 3 pharmaceutical journals. He is pioneer in establishing pharma education in Uttarakhand and has the distinction of organizing states' first national and international seminar in pharmaceutical sciences. He has published extensively international and national journals. He has organized, actively participated in national and international conferences by means of lectures, poster evaluator, chair session, and invited speaker. Presently, he is guiding students for PhD enrolled in UTU, Dehradun, and Kumaon University. He has received various awards and honors. Acharya Narendra Dev Alankar awarded by Acharya Narendra Dev Shiksha Nidhi, Uttar Pradesh, and honored for excellent contribution in the field of pharmaceutical research and academics by Social Talk, World Books of Records, Indian Pharmacopoeia Commission during Health and Wellness Conclave held at Dehradun on 20th March 2020. Welcome, sir. I request you sir, to please address the participants.
Thank you, Prerna. And I hope I am audible to all of you. You can hear me?
Yes, sir, you are audible.
Okay. So thanks a lot, Prerna, and all the organizers, Dr. Ashwini and all, for inviting me in this refresher course. We have some good number of participants here. Before proceeding, Prerna, can you tell me, they are from all over India, participants?
Yes, sir. We are all over India, sir. Nagpur, Maharashtra, Haryana, all states, all participants are joining from today's session.
Now, how many of them are comfortable with when in the talk? Sometimes I start using Hindi also. That is my language. We call it mother tongue. So, are they comfortable with that? अगर मैं हिंदी के बीच में बोलूं, तो किसी को कोई दिक्कत तो नहीं रहेगी?
No issue, no issue, sir. You will communicate in English or
Hindi.
Hindi.
The presentation and PowerPoint is in this thing. The language of preference, that is English right now. Okay, but
okay, sir, no issue.
And thanks for the lovely this thing introduction. I thought that will be longer than my presentation. You have been very nice and kind, Prerna. Now,
okay.
All of them are faculty, or there are some researchers also? All of them must be faculty people only.
Sir, all are faculty.
Huh. So see, we should know the target audience also, because if they are faculty, then I have to be more careful. Some of them might be knowing issues better. I think we should never be proud of anything. This is my philosophy. So, in case there is anything, you can correct me at any time. Okay, I do not consider myself on authority to any of the subject. What I am trying to express the information. So, should I share the screen? No.
Yes. Yes, sir. Yes, sir. You can share.
Okay. Maybe this one. Can you? I have joined with 2 systems. So can you see this presentation? आप देख पा रहे हैं?
No, sir, we can not see your presentation.
Can you see the presentation?
No, sir.
Okay, no problem. Actually, I was my another device. Is it?
Now it's visible.
Yes, sir, it's visible.
Okay. So the topic for today's discussion. Whenever our students they start their presentation, they invariably use this language. The topic of my presentation.
Sir, again, presentation is not visible.
It's not visible.
Sir, sorry to disturb you. Again, presentation. No, sir, not visible.
Not visible. Okay. Just give me a minute. Now?
Now visible, sir. Now visible.
Okay. Actually, admin, no problem. Okay.
Okay, sir. Okay, sir.
So, because I'm using another device for the purpose of ease. Okay. So the topic that I have chosen to discuss with you is modern approach to phytochemical analysis of herbs. Basically, we are concentrating on the extraction part. Okay. If there is some modification, the extraction of phytochemicals or herbs is the basic this thing. Now that most of us, or rather all of us are faculty members, we start with the philosophy of education. Okay. And when we talk about
Sir,
philosophy,
please. Sir, please.
pardon?
Hello, sir. Sorry to disturb you. Sir, please call your screen on full screen.
Is it not in the full
Slide
screen?
is not visible on the full screen. No, sir. It's
Okay,
not shown on full screen.
एक मिनट.
Okay.
नहीं आ रही, फुल स्क्रीन में नहीं है. क्यों हो रहा है?
यहाँ लाइट नहीं चलता मेरे को, तुम नीचे ही रहती है।
Wait a minute, हाँ. जहाँ पर स्लाइड है वो नहीं दिख रहा है ना, इसलिए प्रॉब्लम हो रही है. इससे चल जाएगा काम? क्यों भाई? थोड़ा सा प्रॉब्लम है.
गुड मॉर्निंग सर.
हाँ
सर ये बढ़िया सर, बढ़िया सर. ये ऊपर जो स्लाइड शो है ना, वहां क्लिक कीजिए आप.
हाँ, एक मिनट होल्ड करना. ठीक है. जो अभी इससे पहले आ रहा था वो नहीं दिख रहा है?
नहीं सर, अब वो चला गया है. तो वापस से अपनी शेयर स्क्रीन करके ऊपर स्लाइड शो का ऑप्शन है,
मेरी दूसरी डिवाइस जो है वो आपको दिख रही है?
हाँ सर दिख रही है सर. इसमें सर ऊपर देखिए, ऊपर स्लाइड शो लिखा हुआ ना, यहाँ क्लिक कीजिए आप.
हाँ,
ऊपर स्लाइड शो लिखा हुआ सर, सबसे ऊपर चौथे नंबर पे स्लाइड शो लिखा हुआ आपकी स्क्रीन पे.
एक मिनट रुकना. हाँ, जो ये वाला ये नहीं दिख रहा है, जो अभी हम दूसरी
दिख रहा है, पर वो एक्चुअली अभी स्लाइड शो में दिखाएंगे तो पूरा बड़ा दिखेगा. आप ऊपर से होम, इंसर्ट, डिजाइन, एनिमेशन, स्लाइड शो लिखा है, उससे स्लाइड शो पे क्लिक कीजिए सर प्लीज.
ओके, एक मिनट.
ये स्लाइड शो, स्लाइड्स अगला सर, अगला 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 सर, इससे अगला ऊपर स्लाइड शो. नेक्स्ट सर, नेक्स्ट 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 सर. इससे यहाँ सर. यस सर.
ठीक है, ठीक.
हम बिगिनिंग कर दीजिए सर.
कम देखिए, अब आ गया ना? राजी तो क्या?
नहीं सर, अभी नहीं आया.
मैंने फॉर बिगिनिंग भी कर दिया है. क्यों? कहाँ दिक्कत है?
एक सेकंड आ रहा है. सर इसको कट कीजिए. ये जहाँ पे आपने स्लाइड्स राइट लेफ्ट में जो स्लाइड्स है ना सर, यहाँ क्रॉस का निशान है, इसको क्रॉस कर दीजिए नीचे से.
अच्छा, ठीक है, ठीक ठीक.
अब
Is
ठीक
it
है
okay
सर,
now?
यस सर,
Now
यस
it is okay. Okay.
जी सर,
Yes, sir, it's
Okay.
okay.
ये एक्चुअली क्या हो रहा है ना, वो तो आ गया आपका, ठीक है, लेकिन वो नेक्स्ट स्लाइड नहीं दे रहा है। हाँ ठीक है, ठीक, नाउ इट्स ओके, सो वी विल स्टार्ट नाउ। प्रेरणा, यू कैन टेक योर सीट आल्सो, व्हाई डोंट यू सिट डाउन प्रेरणा? ओके,
Yes, sir.
so हाँ, you phytochemical analysis of herbs. In this part we will be talking about the extraction process, mainly modern approaches in phytochemical analysis. Okay, so when we talk about herb, I love to talk about the first thing जो Socrates ने, मैंने आपको अभी just few moments earlier I had talked about Socrates and philosophy. The first thing that Socrates has said, whenever you deal with a situation, what is the definition that you are talking about? आप उसको कैसे define करते हैं? How do you define the situation? That is the first thing. He had talked about concept and perception. Most of the time, we go into perception and not the concept. What to be understood is that what is the actual concept. So to define herb, we go for what is herb actually. So actually herb, technically speaking, is a plant with stems and withers and dies down after flowering. So we can say that it's an annual process, but in common usage, when we talk about herbs, we are actually talking about the plant whose leaves, stems, roots, flowers, or seeds have culinary properties. Okay, they can be used as a medicine or a drug. It comes close to the definition of a drug. Now, the good thing about using the tool,
नाजिया नहीं आई आज, नाजिया नहीं आई।
so how do we define herbal medicine? See, whenever we say herbal medicine, we use a term loosely. When we talk about herbal medicine, we are actually talking in a little loose term. I won't say it is an offensive. When I say loose, it is not an offensive term, but we are not very careful about deciding what is actually herbal medicine. So WHO is very clear about what is herbal medicine. Okay, it says finished labeled medicinal product that contain an active ingredient, aerial or underground part of plant or as a plant preparation. Okay. Plant material also include juices, gums, fatty oil, essential oil, or any other substance of this nature. Herbal medicine may contain excipients. Additionally, in that medicine, you can use excipients. Formulations you will require excipients, so that is not a problem. In addition to the active ingredients, medicines containing plant material combined with chemically defined active substances, including isolated constituents of plants, are not considered to be herbal medicine. So here is the catch. If you look at this, okay, the medicines containing plant material combined with chemically defined active. I'll give you an example of this thing. Topical painkiller, this thing, analgesic painkillers, which they use capsicum with diclofenac. आपको मूव वगैरह में कैप्सिसिन मिल जाएगा. So now this is not a herbal preparation because we have already added synthetic or semi-synthetic product along with the natural product capsicum. Similarly, atropine, when we say its origin is herbal or natural, but it is chemically defined substance, so it cannot be called herbal medicine. So this thing has to be kept in your mind, that what are herb and what is herbal medicine. So those when you use the term herb alone, it is not herbal medicine. And when we say herbal medicine, we have a well defined this thing, herbal medicine explanation from WHO. Okay. Now this phytochemistry, when because we are talking pharmacognosy or phytochemistry, they go hand in hand. Phytochemistry, plant chemistry is the study of chemicals found in plant, describing their extraction, isolation, purification, identification, and structural elucidation. Okay. जो हम आर्टिफिशियल बात करते हैं, it has a very specific, unique features. Okay, that's why it has anti-malarial properties. So structure elucidation becomes an important and integral part of phytochemistry. Okay. Now, when we talk about phytochemistry, we have a broad classification. We are all teaching in our class primary metabolites and secondary metabolites. Primary metabolites which are required for the plant, and secondary metabolites which the humans or we use it for our various purposes. Okay. Now, many of you may have questions in your mind why this kind of the classification in secondary metabolites, alkaloids, phenyl, propanoids, polyketides, and terpenoids. See, perhaps this will give you an idea of what I am actually talking about. Okay. You can see we have alkaloids by building by this thing. Block is nitrogen compound. We call it alkaloid when nitrogen is there, alkaloid is there. Another important thing about alkaloid is the significant pharmacological activity. If pharmacological activity is missing, probably it is not an alkaloid. Similarly, we have phenyl propanoids, which are derived from phenylalanine, and polyketides. Polyketides is one very important class of this thing, phytoconstituent. We have given an example, acetate melanoate together combined to give you polyketides. It's a diverse this thing, class of compounds, and very important. We'll see why it is important. First thing, let's look at the aflatoxin B1. Aflatoxins, we know usually they are produced by microorganisms. Here this aflatoxin is carcinogenic in nature. So they are diverse and varied medicinal uses. Macrolides, doxycycline and all. Macrolides were used during this corona also. So they are macrolides, amphotericin, flavonoid, doxycycline, they all belong to polyketides. And then the complicated part of phytochemistry is terpenoids. Okay. They originate from isoprene and they go up from essential oil, simple molecule, to complex natural rubber. Okay. So phytochemistry, when we talk about, it has got all sorts of variations. So this becomes difficult also in that case. Okay. Similarly, we have some examples of this thing, phytoconstituents and their uses, which is clear from here. Alkaloids, like I said, have a significant pharmacological activity. Okay. Socrates, I was talking about, he was given an alkaloidal this thing, poison. He was poisoned, Socrates. Okay. So that was, and Socrates उसका नाम ही रख दिया गया. Okay, just because Socrates was poisoned with that alkaloid. So there's different, and one thing that you must all keep in mind at this point, why herbal medicine, the definition, when we are talking about the definition, they have said a individual constituent, if it is identified, is not an herbal medicine. It has to be a mixture of constituents because synergy plays an important. This is not very well explored part of pharmacognosy, but it should have been explored. And this thing, synergy plays one very important role. And this is in Ayurveda, you will see there is rarely any individual herb that is being used in formulation. Usually it has a combination, and many of them they are producing only synergy. They may not be pharmaceutically active as such, but they are assisting the other phytoconstituents in their this thing, activity. Okay, so if I say you are saying you may have this question in your mind that antiviral, antimicrobial, these are the activities that are given. This is just to let you make up your mind. It is not that alkaloids are only antimicrobial, they are only antimicrobial. It is the overall effect of the extract. It may be oil or anything. Okay? Now, another important thing that has come up now. We all talk, maybe you might have heard of lecture in green chemistry. Nowadays, then we talk about green chemistry. And there is an upsurge of interest in clean energy exchange and material conserving processes, which is an entirely new discipline. Process intensification, it has become focus of large and sustained effort all over the world. We are talking about process intensification. I will come to this slide when we will talk about extraction, but right now you must understand that process intensification. It has not become, it has not remained a very simple process. You go to a substrate, you extract, and then that was you get an extract and you use it. It's not any chemical engineering development that leads to a substantially smaller, cleaner, and more energy efficient technology. Process intensification में आप क्या करना? What you are doing here is you are smaller. You are trying to use a compact this thing process which is cleaner and energy efficient. Okay, we are this thing. Green energy now is the keyword. Solar energy is being used, and that's why we have now self-reliance in the case of energy sector also. So energy efficient technology is another keyword for the future. Okay. Now when we talk about phytochemical investigation, the systemic investigation of plant material or phytochemical involved, there are 4 different stages. When we talk about phytochemical, the procurement and quality assessment. The first thing we are teaching this thing in pharmacognosy also, quality of the, we have a separate chapter in pharmacognosy, the procurement and quality assessment. Because when we talk about synthetic medicine or the active pharmaceutical ingredients, it is well defined and easy. But when we talk about quality assessment of raw material in herbal medicine, it is a complex process. Okay? It starts with extraction, purification, and characterization of the constituents of pharmaceutical interest, and then there is in process quality control. Then you have another parameter, investigation of biosynthetic pathways, and then quantitative evaluation. Okay, and for this, what do we need? These are again paper chromatography. That is the most basic technique. You have thin layer chromatography. In thin layer chromatography, we have a variation of preparative TLC, gas liquid chromatography, GLC, HPLC, high performance. Now, as the name suggests, high pressure. From high pressure, it changed into high performance. This technique was so effective that from high pressure to high performance liquid chromatography, it was renamed very Christian. Then you have column chromatography, that still remains the basic of the research in phytochemistry. Then there are various modifications of chromatography, gel permeation chromatography, affinity chromatography. Okay. Now, then what we do is we go for a method of identification. You all are teaching in your class detection of alkaloids. You have various reagents for this thing. So this is just for brushing up or kind of recap. This isn't something that you have to be taught. Almost all of us are familiar with these detection of tannins, phytosterols, and all those sulfur. I mean, this is basic this thing, pharmacy. Now then we go for quantitative estimation, which includes total tannins, total sugars, total starch residue, phenolic contents, and total flavonoids. This is basic quantitative estimation when we talk about phytochemistry of the natural product. Okay. And then we have extraction that we should understand if we are about to understand phytochemistry. Okay. Extraction is the crucial first step in the analysis of herbal medicine because it is necessary to extract the desired chemical component from the part material for further separation. जब हम extract करेंगे, that is the first step. Whatever you have to do, you require the basic raw material. You can say this is kind of getting the raw material for you. So what process that we are having in extraction is transference of compounds from a solid or liquid into a different solvent. Okay. Now the word solid or liquid is being used here. Okay. And then you have selective removal of components from a mixture. They may move from liquid to another relatively soluble this thing, liquid. Okay. Then you have another important aspect is standardized extraction procedure for food, drug, medicinal plant part, what we call it, is obtained from the therapeutically desired portion and to eliminate unwanted material. When you go for extraction, first thing you do is you do defatting. Non-polar, polar, we go. So basic procedure is removing the fat. Defatting हम तो अभी use करते हैं उसके लिए because that is unwanted part of the extract. So eliminate unwanted material with selective method. Okay. Now, this is the slide that I wanted to discuss with you. When I had earlier told about, I'll come to this slide, which is of importance. Earlier, the classic procedure was the scientists were interested in yield of extraction, various types of metabolites that we get, and there was some selectivity. Okay. But now, today we are having a different this thing ideology when we talk about this thing phytochemistry. Okay. Initially, I had talked about the process intensification, and this is here you will see the difference between the conventional or classical extraction and the process intensification. Okay, we are using reduced amount of solvent. Minimum unit operations are performed for extraction. Then we have reduction of extraction time. Okay, uses of renewable plant resources. This is another important way. Even the plants in the modern day also. Okay, they have become endangered just because we had not thought about renewable sources or resources. Then security and safety. You see, these points are missing in earlier work. There was not much consideration was given to these points. Environmental impact. again, this green chemistry has now become a part. I don't know how many of you are practicing, but green chemistry as a pharmacist, chemistry is an important and integral part. You cannot avoid chemistry, and if you cannot avoid chemistry, you have to know the green chemistry. And finally, the commercial aspect. Whenever I talk about pharmacognosy, the definition I have to give to my student is study of biology, biochemistry, and commerce of the natural products. Okay. You can use the term drug also in case of some this thing, herbal medicines. Oh, sorry, pharmacognosy. Drug is the anything that is obtained from natural product, and it has diagnostic or therapeutic value. So rapid return of interest is very important. If you are not thinking in commercial terms, justification to your act of education or research, anything like that. Rapid return of investment. Nowadays we talk about publish and perish, and this thing patent and flourish. So patenting has become an important part of the research. You just don't publish your research, but try to get it patented. Try to do the research which has some commercial value. So you will see that we have different parameters as on date as earlier. Intensification has now become a keyword. Okay. So this is the conventional procedure for successive solvent extraction. The plant material, it is extracted with various types of extract to get this thing, desired chemical constituent. And in this, the problem that usually is faced with the alkaloids. Okay, so we have a different set of filtration process or extraction process for alkaloids, where they are basically basic in the alkaloids. They remain acid molecules attached with acid molecules, so they have to first be basified. They are basic in nature, but ammonia is the most commonly used, and it is good enough. Ammonia to separate it out and then have it in some organic solvent. Okay, and then you can further separate alkaloids. Again, a routine process. So we are not going into the details of this. Wherever it is required, we'll talk about it. Now, when we talk about this thing, extraction, okay, like I said, we have different types of solvents. These are commonly used with this thing. Maybe ethyl acetate, one of the commonly used solvent is maybe missing, but this will give you a broad idea of what is the choice of solvent when we are talking about extraction. There is no uniform solvent for any of the chemical compounds. Like I said, alkaloids, they are organic plant acids. Okay. You see, there are every day we start our day with this thing, a cup of tea to get refreshed. What it contains is alkaloid, and what the process we are using here is decoction. Water soluble extractives are being used, and alkaloid becomes alkaloid gets extracted here. Okay. So you will see it's in water, ethanol, chloroform, even in ether. Okay, it is diverse. These chemical constituents, they are diverse in nature, so you cannot have a uniform this thing. There is no rule of thumb. When we talk about extraction, You cannot say if it's methanol, all the alkaloid will come only in methanol and it may not come in ether. You don't have that this thing. Okay. Now these are the common processes that we are doing in our lab. Maceration, infusion, percolation, digestion, decoction, hot continuous chocolate. I don't think anyone who has done pharmacy is not familiar with chocolate. Every lab, almost it may be chemistry, pharmaceutical, pharmacognostic, chocolate is required. Okay, so chocolate extraction is most common, just like TLC. TLC, chocolate, these are common procedures. Okay, then your hydrodistillation, water is used. Water may be used in combination with ethanol. Hydro alcoholic extracts are commonly used. Okay, many times the compound is thermolabile, where you cannot heat it above a certain degree of temperature, whereas in chocolate you have hot continuous extraction. So you may use hydroalcoholic extraction there. Okay. Now, when we talk about the extract, we are actually talking about the basic parameters that influence the quality of an extract. Unlike, like I said, what we are talking about API. API are simpler, easier. Now here in natural products, the basic parameters are different. Plant parts used are as starting materials. Okay. We use the solvent for extraction, and there is a extraction procedure. Okay. Now the nature of the plant material. This is phytochemicals. They have, like we have just seen, they are diverse. So the nature of plant material and the kind of this thing constituent they are going to extract becomes important. Okay. The degree of processing, moisture content, particle size, they all have effect on the this thing, the final product. Okay. Then you have type of extraction, time of extraction, how long. When we proceed into this presentation, we will see time of extraction, how it is important and how it can be reduced by using different techniques. Okay. Another method of extraction, when we talk about variation in extraction method, usually depend upon length of the extraction period, the solvent that you are using. We have seen a list of this thing, solvents. Okay, so the solvent that you are using, the pH of the solvent, temperature, particle size of the plant tissue. Size reduction is equally important when we talk about the extraction of drugs. Okay, and then solvent to sample ratio. You cannot just go on adding the solvent. Okay, because again removing of solvent will become a problem for you. So solvent to sample ratio is always very critical and important. Okay, so when we are talking about this thing, extraction and its significance, like I said, no, concept and percept, there are two words we are talking about. What we perceive, what is the really seen, and what actually is the situation are two different things. Okay. In normal process, when any drug, now I'm talking about any drug, we are not limited to this thing, natural products. If you add it into suppose we take a solid liquid interaction, you see actually that extract that is available to you is shown in dotted lines, whereas a large part of the solid state remains undissolved in solvent. And here we are trying to see that how can this, normally this is the normal process. You add a solvent, you don't do anything, simply percolation, maturation or technique. The large part of it may remain in the menstruum or the solid state. So we are trying here to increase this, reduce this inner circle, so that we get the maximum yield out of the extraction. Okay. So for that, we have size reduction, extraction. These are various steps which are involved in extraction of this thing, medicinal plant. Okay. Latest techniques like spray drying. We have what we do in lab. The most common thing is you take this thing, extract, keep it on a water bath. But this thing, spray drying, solvent extraction, cold percolation, concentration, there are various techniques that are being used nowadays. Okay. The list of extraction, we will not go into detail of all of these. Our discussion will remain focused on sonication and microwave assisted extraction. There are many. If we go into these extractions, there will be a lot of time required. Okay. So we'll focus on sonication. I'll give you just an idea of what these extractions are. These are rather not being used very commonly in our labs. Okay. Now, sonication has become a common this thing, extraction procedure. Okay. We have been teaching many procedures, but sonication extraction or ultrasonic assisted extraction, UAE is the term that is being used for extraction of these plant materials. Here what we do is the use of ultrasound waves is done to enhance the extraction procedure. Now, what happens is here is in conventional this thing, extraction process, diffusion is the main the process through which the drug is leached out or pulled out of the cell. But here, when we use sonication of microwave, this thing, ultrasound, what you are doing is you are able to cause cell disruption, which causes the release of your active constituent. Okay? This gives you a better yield in a shorter time. So you are also able to shorten the duration. Okay. We have discussed about the time, importance of time. Conventional extraction, we must have done this thing, extraction using software apparatus. Okay. If you are using simple percolation technique also, then you go with imbibition, you keep it in the solvent for a long period of time, and it takes hours. Okay. This is not the case with the UAE or ultrasound assisted extraction. Now, there are various parameters that are to be used: extraction temperature, extraction time, solvent to solid ratio, and then important thing about ultrasound is the amplitude of the sonication is 20,000 or more vibrations per second, and the frequency that is being used is in two different types of range frequency. It may be high from 2 to 21 megahertz and as low as 20 to 100 kilohertz. And this causes lysis and inactivation of the cells. Okay. Now this figure will tell you about the this thing.
ਉਹਦਾ ਟਾਈਮ ਬਰਬਾਦ ਕਰਨਾ ਸਾਰਾ ਕੰਮ ਉਹਦੇ ਨਾਲ ਪਾਉਣਾ
The instrument that is being used for ultrasound extraction. Okay. You have a unit which produces ultrasound or generate ultrasound, and these ultrasounds are transferred into the solvent through ultrasonic flow. Okay. Otherwise, apart from this, sonication machine, this is a simpler unit, as you can see. Okay. This is more simplified version of the same the figure that we have just seen before this slide. There is an ultrasound generator, there is a chamber and ultrasound probe which transfers the sonication into the sample, and water bath is required to control the temperature. Okay. Then you have microwave assisted extraction. Now this is a process of using microwave energy to heat solvent in contact with the sample in order to partition analytes from the sample matrix into the solvent. May sound a little complicated to you. Okay. Transfer of analyte from the sample matrix into the sample. It simply means how the sample will be diffused or brought into the solvent. Okay. Now microwave nowadays has become a very common thing, apparatus or equipment. It's a household equipment. You go to any eatery and you will find microwave there. Okay. You ask for anything, and अब तो रेलवे स्टेशन में आप जाए तो वहां पर वो पिज्जा 2 मिनट में माइक्रोवेव में गर्म करके देता है आपको. Okay. So what is the microwave conventional heating and microwave? There is a difference. Okay. Conventional heating requires a lot of energy, time, and if not done in controlled manner, it can be difficult. Whereas with microwave heating, you have reaction rate acceleration, milder reaction conditions, higher chemical yield, lower energy usage, different reaction selectivity. This will give you the idea about what I was talking about when we were saying efficiency and intensive use. Microwave oven uses 0.36% of kilowatt per hour of energy as compared to convection oven. Okay. And electric oven, it starts with 2 kilowatt. अगर आप एक इलेक्ट्रिक ओवन घर में यूज कर रहे हैं, इलेक्ट्रिक मोटर, most of them they use energy in the range of 2 kilowatt. Okay. And if you have a gas oven, the energy use goes to even higher side. And see, we have talked about the commerce. You can see here that the microwave oven cost wise is most effective. Okay. So industrial aspect, if you are doing any work, any research, industrial aspect has to be kept in mind. Okay. Then we have two general effects: specific microwave effect and non-thermal microwave effect. Okay. Specific microwave effect, where the heat becomes a primary condition, heating, whereas non-thermal microwave, you don't use that much of energy is not required. Extraction becomes major important. Now, this slide, I think, yes, you can see the difference in the yield. Okay. Microwave assisted extraction, the yield of aniline is increased from 1.25 to 1.86%. And when we are talking about the volatile constituents, this is a significant increase in the chemical constituent. Then this is of superior this thing. Superior quality also. Okay. The thing that you are getting in is of superior quality, percentage yield is also increased. Okay. Now there are various solvents sometimes used, which may be dimethyl sulfuran. For these two, these are not very conventional solvents. That's why I have given the diagram. DMSO, ethanol acetic acid. These are day-to-day chemicals. Okay. Then you have multiple head space extraction. Okay. Now, here what we are doing is we are using a passive means. We are not using any, usually no force is applied. It's simply whatever is there, just check it out. Okay. Now let's see what we are talking about. We generally take the volatile and try to absorb it on the some suitable material. Okay. When you will see the figure, you will understand. Right now there are two types of this thing. Static head space. In this, there is nothing doing. Simply take a sample and check it out. Okay. Whereas in dynamic head space, the environment is repeatedly by using some gas or some continuous gas extraction process, where you keep on changing the environment also. Okay. So you add some gas or something like that, so it becomes dynamic. Otherwise, it's static. There is a solvent and this thing, a saturation, and you take the sample. Okay. Okay. Now, we were talking about micro distillation. This is a simple instrument. You can see micro distillation, a small amount. This solid, this thing that we were talking about, solid phase micro extraction. Okay, head space extraction. These are combined units. Now, if you look at this figure, head space solid extraction, you will understand this thing. You have this part of your sample chamber. It contains the saturated vapors. Okay, this is extraction of volatile materials. Okay, and this is the absorbent surface. You take the sample simply through this. There is an injector needle, and here you can see the conventional, we were saying, where you take the sample, solid and liquid phase, sample is there and the vapor is produced. You simply static, where there is no, this is this cap, this has a diaphragm which can be punctured just like any injectable, this thing, multi-storage device for injectables. Okay. एम्पूल और वाइल आपने पढ़ा है, जो वाइल होता है, what is it? It has multiple doses. So what nowadays it is not being used regularly, but earlier आपने अपने बचपन में बहुत से लोगों ने देखा होगा कि there was a vial and they will insert a needle. Similar process is being used here. Okay, once it is sealed, enough of vapor is produced and that vapor is collected. Okay, no heating, nothing. आप कोई भी एक्सटर्नल सोर्स नहीं ये कर रहे हो. स्टैटिक और डायनेमिक में ओनली डिफरेंस इज दैट यू pump in some gas so that higher amount of volatile constituent is this thing. But this gas has usually is non-reactive. It does not react with solvent or the solute. The idea is to increase the amount of sample that is to be collected. Okay. See, this is what the plunger is actually about. ठीक? This needle helps you in collecting the this thing sample from the vial that we had just seen. Okay. जो हमारा इसमें आप ये चीज देख रहे हैं इसका detailed version. This is the detailed version. You have a plunger, okay, and this hub window, usually you know what is going around inside. Okay. And this is how the analysis is done when we talk about the headspace extraction. You have a sample holder, whatever is pulled out through the plunger into the sampling chamber, this sample is withdrawn and placed into GC injector port, and through gas chromatography you find out what is the this thing. Okay. Your active constituent is there. Food industry, this is frequently being used. Okay. In pharma, we are still industrial level, they are using it, but in food industry, this process is being used intensively. Okay. So I think it is already 11:16. Time to say thanks. But before this, you can see this thing, various types of adsorbent that are being used for this thing, headspace extraction. Okay. I don't have to read this out. I hope you can see and this thing. This is just for your viewing and learning. Okay. So I hope this presentation was meaningful to you guys. आपको इसमें था कुछ तो जरूर देखने और सोचने को मिला होगा. Okay. So thanks a lot all of you for your patience listening, and I will hand over this presentation mic to Prerna again. Dr. Prerna.
Hope you all make the most of this wonderful learning opportunity. Thank you, sir. Sir, your session is very informative. You were very well explained about extraction processes, various informative session and various type of extraction processes. Very well explained, sir. Thank you so much, sir. Participants, if you have any question, you should ask and mention in chat box also.
We can have some discussion also. It's not necessarily it has to be because we
Yes,
all
sir,
are
if any
teachers.
participants.
We all are teachers. We can share our views also on these kind of presentations. And at the same time, if any of you have any suggestion regarding my subject, my specialization is in pharmacognosy. Even that is welcome. We rarely get some 61, including myself, there are 61 participants. So if we have 6 views on profession, it will be excellent. But anyway, let's start. ठीक है, तो shall we call it a day now? Actually, I have some assignments also. तो अगर आप लोग के पास क्वेश्चन नहीं है तो हम आप सबको धन्यवाद कहना चाहेंगे. You have been very kind and you
Sir, please
listen to me.
chat box. Question are in sir chat box.
हाँ बताएं.
Sir, there is one question that how can we decide alternative solvent systems for TLC?
Alternative solvent system for
We
TLC.
don't have a particular solvent like in the literature. It is
Okay,
written toluene.
this is a very common this thing for a new researcher. We have seen this problem. Whenever you go for TLC, the solvent system, but you do not have, like I said, set of rules. Okay, depending upon your chemical constituent, from polar to non-polar, you have to go, and it is basically hit and trial. Okay, you have to go through the literature and you have to find it out on your by yourself. There are well-defined set of this thing solvent systems which are used in TLC. You have to rely on that, and like in research we said it is 99% perspiration and 1% inspiration. So if you are going for a new this thing TLC method for a compound, you have to rely on the conventional reported TLC systems as well as the nature of your this thing. इसमें आपको थोड़ी सी मेहनत करनी पड़ेगी, थोड़ा पढ़ना तो पड़ेगा ही. Okay, so there is no one, at least I don't know any person who will simply you say कि sir this is the plant, these are the types of compounds. So anyone cannot suggest you a particular this thing solvent system. Okay, but yes, there are literature that are available that will help you. Like I had given you a slide where the solvents systems were used where alkaloid is coming into picture, flavonoids, they are getting dissolved in some, so because basically solvent system you are using the separation techniques. Okay, saturation from one solvent system to another solvent system. So what you have to do is to the nature of your constituent. Okay, there is no shortcut when it comes to TLC. Dr. Prerna is smiling. She must be understanding. She must have faced these kind of problems herself also. Okay. Yes, please.
Sir, we should, if we want to select an alternative solvent, then polarity may be considered as a factor.
That's true. Okay, because the nature of constituent is important. Okay, आपको जो मैंने स्लाइड दिखाई थी, अगर आपको याद हो, okay, so various this thing solvent systems like with this क्या नाम लेते हैं उसको पेट्रोलियम ईथर. हम लोग जब डिफैटिंग की बात करते हैं तो पेट्रोलियम ईथर ही कहते हैं. Okay, or n-hexane. So you know, if you are into this thing essential oil, you have a set of this thing polar and non-polar, dichloromethane, this thing chloroform, dichloromethane. You can take these kind of this thing. So एक set of broader I can say the set is existing, but that you have to look according to the type of constituents. Okay. Yes, please.
Okay, thank you.
Thank you, sir. Thank you so much, sir. You will very well explained, and maybe participants are sure about his answers and very well explained. Thank you so much, sir. And now, as a token of respect and appreciation, on behalf of Guru Gobind Singh College of Pharmacy, I would present a certificate. It's on the screen.
Okay, thanks a lot. It
Thank
was
you so much,
wonderful,
sir.
and it is good to have an interaction. Have a good day, all of you.
Thank you so much. Thank you.
I can leave now.
Yes, sir, you can leave.
Okay, thanks a lot. Huh?
Thanks, Lordship. A very good morning to all of you. I once again welcome you all for the second technical session. I welcome you, sir. It's our privilege to welcome Dr. Dipendra Singh, Member PCI, Assistant Professor, Pandit Ravi Shankar Shukla University, Raipur. Dr. Dipendra Singh is presently working at University Institute of Pharmacy, Pandit Ravi Shankar Shukla University, Raipur, India, one of the oldest and biggest university in the state. He is the member of Pharmacy Council of India, New Delhi. He has around 18 years of teaching and research experience. His prime research area include formulation of multiple bioactive delivery system like multi-phase hydrogels, nanosystem, microspheres for wound management, designing strategies for protein and peptide delivery, advanced statistical and mathematic modeling methods, tissue culture techniques, and designing novel system for delivery of plant active and extract. He has published more than 90 research and review papers in national and international journals with H-index 26 and index 58 with citation of more than 3,190. He has completed several projects funded by UGC, AICTE, ICMR, TST. He is recipient of prestigious Department of Health Sciences ICMR Young Indian Biomedical Scientist Award 2019, UGC Raman International Award 2014, and UGC Research Award 2012 to 2014 by University Grant Commission, New Delhi. He is also member of prestigious awards at state level for excellence in research with Young Scientist Award of the State of Chhattisgarh 2006 in Medical Sciences. He is state evaluator and mentor for National Children's Science Congress. He is a life member of various professional bodies like Indian Pharmaceutical Association IPA, Association of Pharmacy Teachers of India APTI, Indian Society of Pharmacognosy ISP, and serving as President Chhattisgarh State Branch APTI 2013, elected Secretary. Chhattisgarh State of Branch IPA. He had been visiting scientist at National Center for Natural Product Research Schools of Pharmacy, University of Mississippi, USA, and Schools of Medical Sciences, Deccan University, Australia. Welcome you, sir.
For a generous introduction. At the outset, I'm overwhelmed, and thank you for inviting me in this STP, AICTE-ISTE sponsored STP program. I'm very thankful to Dr. Aswini, Dr. Kumar, that they contacted me for such type of activity. Though I always say that still I'm a learner. So today I'll be talking about on designing and experimentation. My just to start, before starting, just a few days back when Dr. Aswini contacted me, just I was asking who will be the audience and how we are going to present. So friends, basically, as I'm a teacher and I'm working in this area, and there is always a problem when I was a student, when I started my career, when I was planning for your research. So there is always a problem, or always a question on that how to start with, how to effectively use your energy for the experimentation. So today I'll be going to I'll be sharing some thoughts of my personal thoughts with you regarding the designing of what is research, what are the variables, how you have to design your research, what the areas in which you can explore, or what are the various softwares by which you can maximize your effort with minimum efforts, or you can say with smart effort, how you can get the maximum out of that. So this will be my talk. So I'll be sharing my presentation with you. So once again, I'm very grateful to your Dr. Kumar, who has invited me for this program, and Dr. Aswini, who is continuously in touch with me and just reminding every time me that you have to give your talk on that time. So thank you so much. So I'll be sharing my presentation. Please confirm once it will be visible to you all. My presentation is visible. Is it visible to you all?
Yes, sir, it's visible.
Okay, fine. Thank you. So just this is my presentation which I'm sharing with you. So I'll be talking on the pharmaceutical research designing and approaches in the experimentation. So this is why I was talking about the theme which you have selected. It is a very relevant theme, specifically from the point of view of new faculties and researchers who are entering in or who are opting as a career as a researcher, or as a you can say as an academician, because research is always back to back, it is going with the academics. So I'll be sharing you with few of my experiences and thoughts with that. So basically, if you'll see research, it is from the prospects of if you'll ask people, it is different from different prospects. If you'll ask from the point of view of a children, or from a point of view of a student and who is learning in school, So they are having a different perspective for the research. When you'll talk about when a faculty, then it is a prospect changes. When you'll talk in reference to the industry, again the research, the purpose of the research or the definition changes accordingly. So right now you have seen from the government of India, from the state government, it is always they are emphasizing us to become self-reliance or so just they are putting effort to initiate the thoughts, innovative ideas, what we are having, how we can improve, how we can come with the indigenous systems or research research which will lead to the product development, finally making us self-reliance. So research, if you talk about research, it is a dynamic. Everybody is having a different prospect for this. But in short, when I was going through the literature or when I was preparing for this presentation, I got this a line which is given by the Tuckman in 1978. So that was just, it clicked me. So I was willing to share, because if you talk about research, everybody is understanding what is research. You all have your understanding of research. But in a single line, he has defined, or he has given a systematic means of problem solving is research. Exactly, if you'll see the end, what the end you are expecting from that, always you are having some target, and through the various processes of research you are trying to get it. So that is basically it is a systematic way of doing it. So if I'll define research, that which involves testing or attempting to prove a hypothesis by a way of experimentation. How just we are expecting something, we are giving a hypothesis, and the way by which you are trying to prove your hypothesis, that can be concluded as a whole process of research. So research, when you are if you are involving in research, then planning is very important. If I'll talk you about, maybe not about the research, but maybe if you are planning for the day-to-day work, if you will not have a systematic plan or path for it, you will end nowhere. जो मैं यहाँ आपसे जो शेयर करना चाहता हूँ कि चाहे आप घर के डे टू डे के जो काम है, प्रोसेसेस हैं, टीचिंग के असाइनमेंट्स हैं, आपके क्लासेस हैं, आपके नॉर्मल प्रैक्टिकल्स हैं, वहां पे भी हमको एक जो प्लानिंग है, दैट इज वेरी इंपॉर्टेंट. अदरवाइज यू आर मेकिंग एफर्ट्स, you are putting your energy, you are doing so much of work, but what the result or what the output which is required from that whole effort, it will not equal to the what the effort you are putting in. So same is with the research. Planning is very important. Otherwise you will end up with a puzzled situation in which you will be in situation that where to go, how to go. I have made so many effort, but still I'm not getting anything out of that. So that situation comes, or that is a very frustrating situation when we start with a new research. So because of that, planning is very important. Adopting a system is very important. Otherwise it will you will end up with the some conclusion that I'm having a very difficult experiment, or I am not getting away. There are so many ways where to go. That is, or that will again you will end up with a dreadful experiment that this is very I'm not getting it. It is very dreadful, and you are hitting your head there. So this is just one cartoon I'm sharing with you. Ki normally this happens with us. Ki when something, it is very easy to say that to work for a new topic working on a topic where the literature is limited. You are among the frontline runners in that area. So at that time it becomes very tedious to work. So this is how systematically we have to proceed. So ultimately you will end up with the burden of experimentation or research. It is called burden of experimentation or research, that what it is expected, the performance is expected, you are definitely you will lag there. And जो आप अल्टीमेटली जो हमसे जो एक्सपेक्ट, जो हम जिस चीज का हमको माइलेज लेना था, जिस चीज पे हमको कैपिटलाइज करना था, वो आपके ऊपर बैठ के घूमेंगे. जो रिसर्च से आपको पेस मिलना है, जो रिसर्च से आपको प्रॉस्पेरिटी मिलनी है, वो आपके लिए एक बर्डन जैसा हो जाएगा कि हाउ टू परफॉर्म इट. सो दैट इज अ वेरी सिचुएशन फ्रॉम विच वी हैव to counter it properly, or to plan it properly, to execute it properly. So basically, if I'll talk about the research, these are the basically 5 very important points which is expected from the research. If you are planning for the research, that it should be systematic. That I'm talking about the various.
Sir, your slides are not moving.
Okay, just please.
And sir, full view of the slide is also not visible.
Just I'm again I'm discontinuing. I'm again sharing with you.
Okay,
So just
okay,
please
okay.
confirm me once again. Okay.
Okay, okay.
Right now my slide is visible.
So it's now also same thing is happening. Means full view is not there.
Okay.
So go to the slideshow.
No, I'm doing it, but it is not appearing there. So let me do it again once again. I'm again stopping the share, and again I'm re-sharing it. Okay.
Okay.
Please confirm me once it is shared. Is it visible? Full slide? My slide is moving?
No, sir.
It's not visible, sir.
It's not.
I don't know what the problem is there. Actually, I have shared it again, and in my system, it is moving.
Yes, sir.
Just check it once again.
Now it's visible, sir.
It is moving.
Full view is still not. So full view is still not there.
It's not moving, sir.
Actually,
It's
when I am
moving.
doing on a full view mode, again it is stop moving. So just again, I am trying to do it once again in a full view mode. You just confirm it is coming.
Okay.
Now it is full view mode.
No, sir.
It is moving.
No, sir.
I
It
think
is
there is
moving.
some issue with my system, and it is not
But sir,
moving
the
properly.
contents are visible. There's no problem.
No, okay. So let me explain. I'll be, if any will be anything will be there. You just in between you give your query, so I'll clear you if something will be not visible there. Okay.
Sir, excuse me, sir. If is it possible, you please run the slideshow. It will
Slide.
slideshow. Right top menu bars are there. Slideshow bar option is there.
Slideshow. Okay. Okay. Let me check with this.
Next, sir. Next one, sir. Next, sir.
After animation, sir. Next, yeah, this is.
From beginning, sir.
From beginning. Now it is working.
No. That's never new, sir.
It is working.
No, sir.
I think so. There is some issue with my system because okay, or I don't know. It is with Zoom because I'm doing after so long. I'm doing with Zoom.
Sir, it's okay, but the contents are visible totally. So
Okay,
there's no problem.
Okay. So I'll be explaining you. So I don't think so we will have any problem with that. Okay.
Yes,
So
yes.
just you tell me when something is there. Just interrupt me. So I'll be clearing you. Okay.
Okay.
So just I was talking about the key experiments, or just what are the key features when we are planning for a research, when we are working with research, which is required. Then it should be systematic. The process, what you are selecting, you should opt a very systematic process. It should not be random while you are working with the process. Along with that, your decided research, it should be logical. It should be I think so the word is very clear. What do you mean by logical? Because for example, you are selecting a topic and you are having a category of say active component, and they are designated for a particular class of treatment, or they are used for a certain area in which it is used, and without any logic you are using it for some other activities. So for the sake of research or using illogical taking a topic, it is not advisable, always not advisable. Then again, what the topic or what the research topic you are taking, considering, it should be empirical. Means you should have a sufficient evidence based on which you are planning. That is always related with the your literature, what you are working with, how you have selected the area, how what the base you are creating for performing such type of experiment, or doing such type of research. It is always very so empirical. Again, it should be empirical. Then it should be reductive. Then why it is said that when you go for, and apart from that I'm telling you, when we decide the topic or of a research, then the fate of the research should be decided for what you are planning, for what you are doing it. So like if my target is, okay, to after performing this research, what the result I'll get, I'll be publishing it for some in some journal. So what is your target, where you are planning? So that is again important. Then if you are planning for okay, I'm doing my research and I want to make it patent. Okay, I'm working for the product or I am working for the process. Again, these type of questions, it should be asked when you are deciding your proposal. At the time when you are deciding your research, it should be clear at that time for what purpose you are targeting your research. Then again, it is expected that it should be replicable. The methodology, the process, what you are opting, it is replicable. Here you consider the thing, it is not a repeating of the things, it is replicable, reproducible. What the experiment you are planning or performing, it should be reproducible to give importance to your research. So there is a great difference between your repeating and replicable. When we are talking about replicable, then I'm talking about the reproducibility of the research methods. So what the methodology you are selecting for the research, it should be replicable. It should be when at any time of set of time, when I am repeating the or when I am performing the complete experiment, it should be replicable. So this is the 5 very key important. Apart from that, if you'll see from the different prospect, people are having different prospects. When they will talk, they will give you so many USP, or USP is further required. But these are few very important USPs of the research which is expected. Then comes the your review, that is whole research process. So it starts with the review of the literature, available information, what the information you are having, what the current status you have. If you have selected the topic, then what is the current status? It is always suggested, or it is always advised that when you are selecting a topic or when you are selecting an area for the research, then why question is every time it is asked. If you have taken a topic, why this topic is taken? So first, always the object should have answer that why I have selected this topic. Then after that, you have to execute that project. Okay, you have saved why. Then you will go to the literature survey, that what the current, what till date, what the work already performed in that area. कहीं ऐसा तो नहीं है कि जो टॉपिक मैंने सेलेक्ट किया already worked है, क्योंकि it is always said कि when idea is coming to your mind, then in 1,000 or more than 1,000 minds, that same idea is also coming at the time of. So it is always said the half life of idea is very less. और आपके साथ जब आप किसी टॉपिक के बारे में सोच रहे हैं तो दुनिया में हो सकता है बहुत सारे लोग उसी समय पे उस टॉपिक के बारे में सोच रहे हैं, तो जो आपने आइडिया जनरेट की उसकी हाफ लाइफ बहुत कम होती है। तो आपको जो आइडिया के साथ में, इसीलिए आप स्टिपुलेटेड और लिमिटेड टाइम में यू शुड एग्जीक्यूट इट। तो आप एग्जीक्यूट करते समय जो लिटरेचर, अवेलेबल लिटरेचर है उसका सर्वे करना, जो प्रोडक्ट है, जो आपका प्रोजेक्ट है उसके लिए सफिशिएंट आप जो सपोर्ट्स हैं और जो प्रीवियस स्टडीज हैं, जो अवेलेबल स्टेटस है, ये आपको पता होना चाहिए। देन अगेन आफ्टर डूइंग दैट आई विल फॉर्मुलेट अ क्वेश्चन कि ओके नाउ व्हाट इज माय आई एम गोइंग टू सॉल्व व्हिच क्वेश्चन, so that is formulation of the questions of your research, then selecting the appropriate research design. Okay, to execute what the aim you have selected, to execute that aim, what the appropriate research design you are going to select. Then collect the relevant data, that is after performing or after deciding the selected appropriate research design, you go to the relevant data, what you are getting out of it, then interpret the finding, that is the interpretation of the data or results. And finally, either you will go for the publishing of the finding, or you will go for the patent of the finding, or you can go further for that. Okay, you are working for some area in which you want to publish your complete research profile in a form of book. Again, this can be one target of yours. So depending upon that, you will do it, and this goes on. So this is the whole research process, if you, but it looks very simple when I'm showing you. Okay, there are 4 or 5 steps, it's fine, everybody is getting understanding of that. But actually when we used to perform it, every step is having a number of steps which are involved in that, and it is always very difficult. For example, I'm telling you, if I'll ask about a research review of literature, so people will say, okay, review of literature. If I'll give you, again, if we'll have time, I can talk on review of literature for half an hour. I can talk on review of literature, how you're representing it, what will be the source, how you will say that okay, no, I have on that particular topic, I have seen all the literature which are currently available, or I have seen the maximum number of you can say the maximum number of literature which is associated with my topic. I have seen it, I made the bibliography of that, and again from that bibliography I am having all the associated literature with me. So that is very important. So this is again, if I'll discuss every point, I can discuss for a long time, and the things are very complicated, as it seems very simple, but this is very complicated. So further, if I'll move on, after that comes the research design. Okay, fine, you have decided the topic, and now I'm talking about research design. So research design, again, it is a very dynamic process. Depending upon the type of research what you have decided, you have to opt the design of that. For example, in case of analytical research, you are having different prospects and different modes by which you perform it. You have to go for the review. You have to, after reviewing, you will go for the analysis. It can be philosophical, it can be historical. That is, it is different from the research what we comes in our mind. So I'll be coming to the point when we say research, what we think. So like that, you are having another classification, and this classification, different number of way of classification is there. From the ease of understanding, there are so many different classification. I have opted one because I was having a target of experimental designing, so I have opted one. This can be further, you will see so many different ways of research designing. Like one way of the classification is qualitative and quantitative. Okay, qualitative designing and quantitative designing. So again, the prospects will be different. Maybe the content will be splitted in different way, and you will have a different way of classification of this study. So in case of descriptive research, again, you are having two type of, either you will go for the case study and survey, or in that you are having a cross-sectional study, longitudinal study. For again descriptive research, if I'll go, so they are again having so many part on that for further studies. We are much interested in the experimental research, because we are always associated with this. So we are much interested and aware interested in this area. So if you'll see, experimental design, again, pre-designs, quasi-designs, true designs, and all are possible. These are possible through the various statistical design. We are having a method of doing it and performing it. And normally we used to opt it and it lies in some class, but at the if you'll do it systematically, you will the results, or I can say the output, will be maximum with your efforts. So if I'll define research design, okay, what is research design? So if you'll say the research definition, the research design is a set of advanced decisions that make up the master plan, specifying the method and procedures for collecting and analyzing the needed information. So in short, you can say that it is a planning. Basically, you are planning for your research. And if I'll say why this research design is very important, so it is for good design, it is the first rule is the first rule of good research. That is, for good research design is the first rule of the good research. आप ये statement जो है, अगर आप ये statement को correlate करेंगे, तो एक और आपका बहुत आपको हमेशा बताया जाता है कि plan your work and work the plan. So it is similar to that. When we talk about a design, if you want to go for a good research design, then if you want to go for a good research, then you have to select a good, that much is important, selecting a design for it. So that is very important. And nowadays the things are very simplified. When you'll talk about this designing or selecting the design or studying about the design, lot of material is available. Lot of the companies who are dealing in that, they are supporting you. So this becomes very now it became very easy to select and go for such type of study. So this is the selection or designing is very important. So now what will be the objective of your research design? If we are going for a design or research design, then what should be the objective? So the main objective should be to gain background information and to develop hypothesis. That I have already, it is a process of your if you'll say the doing of it is a process, one of the step of the research. Then to measure the state of variables of interest. You are interested in which variable, what you want to, that's why after the survey, after the making hypothesis, or after the literature survey, where you are interested. So that is very important. If you'll answer these questions, that will simplify your targets, or that will simplify your that will give you a good result out of your research. Then to test the hypothesis that specify the relationship between the two or more variables, that what you have selected. For example, here you are interested in making a microsphere. So again, question, why microsphere? Fine, you given the answer. Then microsphere of what? Then a lot of variable will come. The polymer will be there, the process variable will be there. So I'll be dealing with this. So I am telling you, okay, this is the objective of your research design. So how you are, what are the objectives you are taking, what are the variables you are taking, what the interaction they are going to show. So we have to answer these questions when we are designing. This should be the objective of your design. Then coming to the type of research design, if you'll say, basically we are having three type of major research designs when we talk about the research design. One is the explanatory design, another one is the descriptive design, and another is a casual design. So if I talk about the descriptive explanatory design, normally this is the initial type of research designs. They are initial research designs. So such type of explanatory design is usually conducted at the outset of the research project. When the researcher does not know much about the problem. For example, a pharmaceutical company is willing to launch, or they are deciding about one product. Okay, we have to go for such type of product, but they are not aware about the market, or what the market is existing or present, what is the situation. So before going for such product, we need so much data. So such type of data, normally they are planned and they are classified into the explanatory research, getting that data. So in this, we are gaining the background information. We are defining the various terms which are involved in that, clarifying the problems and what the hypothesis we are going to, what the hypothesis we are drafting. then establishing the research priorities that will fix, okay, that will help you in designing or deciding that. So this is we are doing in explanatory research. If you'll talk about the descriptive research, this is further a higher level of designing, in which we are undertaking to describe the various questions, which is like I asked you who, what, where, when, and how. So that type of question, and with designing you are getting the answers. Such type of data which is collected in that, they are normally classified into the descriptive research. So this is something advanced. In descriptive research, again, we are having a cross-sectional study, and we are having longitudinal studies. So cross-sectional studies, basically they measures the unit for a sample of the population at only one point of time. So you can say we are taking a snapshot. So this is not a over the time, it is at a particular time you are taking sample survey or cross-sectional for studies whose samples are normally drawn in such a way as to be representative of a specific population. At a particular time, we are drawing to such type of cross-sectional studies, or this type of it is part of this experimentation. There is also a longitudinal study in which we are doing repeatedly measures or for the same sample unit for a population over the time. So you can say this is like movies, they involve multiple measurements and over the time. So they are often they are described as a movie of the population. So this is about the longitudinal your studies. Then casual research, maybe the word is not, but most of the time what we are dealing, this is the area in which we are dealing. Normally we are ending up with this, where the experiments are performed and we are doing with the various experiments when relationship is there. So this is the casual research where the experiments are involved. So if coming to the here the experiment, if I'll say experiment, so again we all are aware that what is experiment. हम सब जानते हैं कि experiments क्या होते हैं. Again, if you'll see the point of view of different people, they are having different definitions of experiment. One I have kept here, that an experiment is defined as a manipulating an independent variable to see how it affects a dependent variable while also controlling the effect of additional extraneous variable. So देखिए यहाँ पे just I have involved three terms. Here what I am saying, defining or as a manipulating as an independent variable to see how it affects the dependent variable and while also controlling the effect of extraneous variable. So there are three terms. I'm again defining you. This is the three terms, that is three independent variable. So if I'll talk about independent variables, these are those variables that the researcher has control over and wishes to manipulate. Okay, so these are the independent variable. For example, the level of the expenditure, type of that is, if you are planning for some product design launch of a product, then type of ad, where display, location, method that is launching for a product, what will be the compensating your salesperson, prices, type of product. So these are the independent variable which are under the control of the experimenter. Then after comes the dependent variable. These are those variables that we have a little or no direct control over but strong interest in changing it. जब हम एक्सपेरिमेंट परफॉर्म करते हैं तो हम किन जो उनके रिस्पॉन्सेज हैं वेरिएबल के, वो हम जिसको हम कंट्रोल करना चाहते हैं ताकि हम व्हाट द बेस्ट आउट ऑफ दैट वी कैन आफ्टर मैनिपुलेटिंग इट विल गेट. Like that we are having extraneous variables are those variables that may have some effect on a dependent variable yet not independent, they are not independent variable. So one by one I'll explain you when I'll go through the this point. So these are the dependent and independent variables and extraneous variables. So after that it comes that what is experimental designing. We were talking about just I given you overview of what is research designing, and now I'm coming to the experimental design, with which we are more concerned, or which is directly concerned with our designing of the research or designing of the experiment, which is very important to get the maximum out of it. So if you'll see the experimental design, it is a procedure for devising an experimental setting such as a change in the dependent variable may be attributed solely to the change in an independent variable. So it is the experimental design. Again, you are having here there are three parts. I'll come to that slide also, that we are having an input, we are having a process, and we are having an output. So defining and designing for the whole process, we are taking now, initially we were not taking, but now we are having so many softwares. So we are designing our experiment for the maximum output with smart, that is called the smart working, with the minimum cost, minimum involvement of finance, maximum output which can be drawn from that work. So this is the we are doing it. So what are the various approaches for this experimentation? So there are three approaches: trial and error method, one factor at a time, which is called OFAT, and also the design of experiments. So that is called normally, in short, it is called DOE. So I'll be talking about this. What is the trial and error method? So trial and error method, when we are not knowing, or when we are not having any data, and with our decisions, we are making trial and error, we are trying to get the experiment. Initially it happens so many times, and we are having very good discoveries out of that. But right now it is not advisable when we are in academically we are performing the research. So I have depicted one picture of a kid who is trying to put the shape in the desired box, or it will go inside. So every time he will try, and if he will go to the place where it is fitted, so it will go inside. So this type of approach is called trial and error approach. Then the next approach is called one factor at a time. Here we are studying the effect of one factor on the various result. One by one we are taking. We are not taking any simultaneous studies. So one factor at a time is like, for example, to very simply, if I'll explain you, it is like people are standing in a queue, and they are interested in drawing money, but only one person can be benefited at one time. So it is like that. Okay, you are studying one parameter, so you will get the effect of one parameter, you will have one data related to that, but that will not be an interactive data. For example, I am studying the effect of stirring speed on the size of a microsphere, or a size of any particulate matter. So at a time I am only studying this. Then in the next experiment I studied about the time. Then in a next experiment I studied about the polymer concentration. So okay, I'll have a data of these three parameters at a three different time, but these are not the interactive data. What the impact of stirring time and stirring speed is there, what is the impact of stirring time, stirring speed, and polymer concentration is there, we cannot study by such methods. So these are the various limitations associated with the OFAT, that less optimized solution, as no model can be developed, no interaction studies, as I have told you, there is no interaction studies are there in that. Then lots of experiment and experiments, and expensive, because for every parameter and for that parameter different labels, so you have to perform so many experiments. And when we are doing academic research, then fund is always a limitation, क्योंकि जो हमारे पास resources रहते हैं बहुत limited रहते हैं. For example, if you are working in a natural products, so they are very costly, the standards are very costly. तो हम सबसे कम में एक्सपेरिमेंट को परफॉर्म करके सबसे अच्छा रिजल्ट कैसे निकाल सकते हैं, तो उनके लिए ये भी फिटेड मॉडल नहीं है. एंड देन डज नॉट अलाउ टू स्क्रीनिंग ऑफ मेनी फैक्टर्स. As I have discussed, ki one person can be benefited at one time. So this is again a limitation of the OFAT. Then comes the why design of the experiment is important. This is a very simple experiment. The simple picture I have kept in my slide. maybe it is visible to you. The faces is representing where you want to end with. So if you'll see the research area, so it can be classified into 3 area. This is the knowledge space. This is the another one, the yellow one, which is represented there. It is the design space, and what it is expected or what we want to achieve, that is the central space where it is expected. So by if you are not systematically working, and you will, if you ended with the in the knowledge space, okay, you didn't got any information or the result which is conclusive, so definitely you will be ending with such phase. Then maybe if you made effort, you reach to the design space, so you will end up, okay, I tried on one carefully chosen problem instance, and it must be best treatment. So maybe you are having, but you are not satisfied what you are getting. And third is a happy phase, when you approach properly, design properly, where it was expected, you reach that place, you reach the central space, and you got the result. So you will always end up with the happy phase. One more example I have taken, you all are have heard about the story of elephant and the blind people. So if you'll see, this is related, why I'm telling you, this is about the data. What the result you are getting, a person is giving you, it cannot be representing a whole situation. So results are only valid for the part of the data treatment space you have explored. So for example, here the people who is working on the blind people, he is giving, okay, fine, he reached to the belly area of the elephant, and he is saying it is wall. Like tail, he is saying it is a rope. Like trunk, he is saying it is a snake. So this is why I'm representing you, because you are having a different data are very important here, and how to how it is explored, how overall. So for making any conclusion related to any topic research, the data and its processing is very important. So getting into the knowledge space, getting into the design space, and finally going to the control space is very important to get a good result out of that. Okay, so this is again a planning of a research. So always avoid the missed reasons. Maybe what the best you are having, it is missed because of your planning. So try to plan or try to do the experimental designing, in which you are having a randomized randomized pattern, and all area of the expected area in which you are going to get the results, it should be covered. Okay, so after that, this is a very important slide I am telling you. This is that about the data. So data, a good data is always having a normal distribution, because when you are performing, so normally you will have a bell-shaped curve, and this is representing a normal distribution data. Here, if you see the selected parameters and the results are in an order, but it is always not there. So, but it is expected we should have a normal distribution of data. In that, you are, if you'll see for the data, that a proper location, that is the central tendency of the data, that is mean is very important. Apart from that mean, the variance or standard deviation, that what the result you are getting, it is how far from the mean, that is called the standard deviation. As just I'll come with a story and I'll give you better explanation of that, how these data are very important, and it should not be seen in a, because you cannot work only by taking the mean, or you cannot go with the results what you are getting out of that. So that is very important, the data. And nowadays, if you are not having a normal distribution of the data, the softwares what you are using, they will suggest you, okay, these data is not normal, and process this data to get the normal data. So these are the when we are working with the experimental design, these are the things which will you will encounter. I'm a student of biology, and I was always having a fear for the mathematics, and we when the formula comes, it starts, okay, you will end up, it is not a design, it is not designing, it is a disaster. Okay, so such things comes into mind. So just for simplifying, because when we are planning for, okay, don't go into the formula and everything, but get the understanding out of that, how it is important, how it is important, how you can implement into the research. So here I have given a simple picture to give you understanding of the various the terms what we are getting and which will be used for. Just I have taken a goals soccer over which are gold in a soccer for last 7 games. So these are the data 1, 3, 4, 6, 6, 7, 8. So if I'll analyze this data, so I'll get the mean, because what will be the average? I'll get the mean 5. If you'll see the mode, which is the repeated most repeated figure, that is the mode is 6. If I'll go for the median, which is the middle of the value of the total data which is collected, it is 6. And if I'll talk about the range, so here the total number of range in which I am drawing the data, it is 7. So like that, we are in the experiment, we are using it. Now, how these are by taking one value, it is not sufficient. I'm giving you one story. For example, I have to cross the river, and I have to decide points from which I should cross. So I collected the data, and who has provided me data to cross the river, who provided me 2 mean data. Okay, one, the value of mean is less, and the another value is high. So obviously my selection, for example, my average height is 6 feet, and I'm having 2 paths, and the mean of the depth of 2 paths is 3 feet and 5 feet. Obviously, if I have to select, I'll select the which is the having a mean of less depth. But this is not the right situation. How? This is the another slide I have kept you. For example, here the mean of the depth I got 3 feet and 4 feet. But when I seen for the standard of deviation, means how the data is far from the mean. So in the first place or the first path, I got 2.4. And in the second path, I got 1.2. So here the deviation, the variance is very high. 2.4 variance is very high. So that what the situation or what the idea I was drawing from the mean, it was not right. So mean should not be considered alone. It should be seen with the perspective of deviation. Standard deviation or variance is always important when we are deciding for the data. So again, when you are having a different standard deviation, then I went for to see the actual data, what the actual data is there. So when you will find the actual data here, the why the mean and the variance we are having, because the variation in the data is very high in the site 1. And if you'll see the maximum depth we are having, may the mean is less, but the maximum depth is 8 feet. But in case of the second site, the standard deviation is very less, means the variance is very less, and the maximum height what in from the actual data I achieved, it is 5. So at a first sight, it is looking that we should go for the mean, but it is not the true story. So we have to see the things when we are dealing with the data. We have to see the things with the standard deviation and the if finally you want to do with the, you correlate with the actual data. So this is a way. Now this is a general model of a process or a system, what we use to perform when we are deciding, when we are working with the softwares, when we are working with the experimental designing. So you are having input, what the information you are giving, what the data you are having, what the things you are putting into the process. So the middle part which is shown, it is a process, and finally you will have output. So on the process, again, you are having so many factors which are working on the process. Some of them are control factors, and some are the uncontrolled factors. So when we are planning for the research, you have to care for these all 4 parameters. What the input we are providing, what are the various controllable parameters, what are the various controllable parameters, and what the output you are requiring from that process. So that is very important. And before going to the start of, because if you go for the various processes, so here again you are having a lot of you can say variables from which you have to work. So what the software or what the experimental design is doing with us, so they will limit you. Okay, you want to select the factors, so which are the various very important factors. Then after doing the factors, what are the various label? For example, I have selected some factor. मैंने कोई एक फैक्टर, आप जैसे मैं कह रहा था कि मैंने एक स्टीयरिंग स्पीड एक फैक्टर लिया. अब इस स्टीयरिंग स्पीड के फैक्टर, दिस इज वन फैक्टर विच इज अफेक्टिंग माय आउटपुट वेरी मच. सो अगेन फैक्टर इज डिसाइडेड. अब ऐसे कितने फैक्टर है मेरे पास? 10 फैक्टर है. 10 में से मैंने 3 सबसे इंपॉर्टेंट फैक्टर लिए. इन फैक्टर्स के बाद में मुझे इनके लेवल्स के ऊपर में वर्क करना है. फॉर एग्जांपल, मैंने स्टीयरिंग स्पीड जैसे बोला. अब स्टीयरिंग स्पीड, फॉर स्टीयरिंग स्पीड what the labels will be. Either you want to run on 10 rpm, 20, 30, 40, 50, 60, 100, and you are having n number of options for that. So again, at a point you are doing it. First, you are trying to limit the various factors which are affecting your process and the response or the output of that process. And then what are the various level of that? So we are designing, we are doing all with this in this process. So some very important terminologies which will be associated or which will come or you will interact or you will encounter every time, that is the controlled experiment. It is, as I already told you, when you are planning, controlled experiment means the study which or the treatment which are imposed on the experiment unit in order to observe a response. Here the things you are planning, you are giving the limits. So you are doing in a controlled manner, you are performing your experiment. So this term will come. Then factor, a variable that potentially affect the response, means the which factor which is finally which is giving the maximum on the response. These are the various factors. For example, I've already told you, like temperature, time, chemical composition, or percentage of the various active ingredients, like that. Then treatment, this word term treatment, when I'm talking in this experimentation or planning, I'm talking treatment, a combination of one or more factors. When I'm using it in a combination of factors, then I'm using the term treatment. Then levels, as I shared you, the values of the factor can be taken on. This is the factors. Then if I'm talking about the labels, then the values of the factor I'm talking about. So when we talk about a label, then it is related with the value of the factor which I am taken. Then comes the effect. That effect, how much the main factor is interacting, interaction is there between the factors and influence the mean response. So finally, what the response I'm getting, the mean response pay, what is the effect of that? If I'm writing effect, then I'm indicating the total influence of over the mean response. Then comes the response variable. It is an output which is measured on the observed base, or it is either on the dependent variables or independent variables. So these are the factors. There are some more factors which you will encounter in this, that design space, which I was talking about initially, to get into the real design space. So when we are giving the limits and we are taking that space, that is called the range of values over the factors are to be varied. That is the whole space you are having for the designing of your experiment. Then the design point, the value of the factor at which the experiment is conducted. For example, जो मैंने variables बोला, तो आप किस particular point पे आप उसको design को perform कर रहे हैं, design उस point पे perform कर रहे हैं, that is your design point. Then the response of surface, that is normally it is unknown because what you are going, and that represents the mean response of any given level of the factors in the design space. What the design space we have selected, what the values or what the factors we have decided, and after of that factor, what the labels we have selected. Okay, so after that, what the maximum the effect we will get, it will come for the it will be represented as a response surface. Okay, so again, these are the few more important to understand or experimental designing. These are the few more key terminologies which are normally you will encounter when you are planning. First is your interaction. Interaction, I've already told you, that is talking about the various factors. When we are taking two factors at a time, then how much interaction they are showing. So sometimes they will show the interaction, sometimes they will not show the interaction. But when together, when they are worked and they are interfering or they are showing interaction, so that is very important. term which you will encounter. And interaction, normally if you get a plot, if you are working with the two parameters and you get a parallel plot, means जितने दूर वो दोनों लाइन अपना दो आपने परफॉर्म एक साथ किया और दोनों अगर पैरेलल चल रहे हैं, एक दूसरे से कहीं एक दूसरे के पास नहीं आ रहे, means they are indicating there is no relationship or there is no interaction between that points or that factors. Or like that, if you are having slope between them, then the maximum slope you will get, the maximum degree of interaction it is showing. So interaction is the term you will get. So interaction हमारा यहाँ जो इसका मतलब है फैक्टर्स से रिलेटेड है कि जो फैक्टर्स हम एक साथ स्टडी कर रहे हैं वो इंटरेक्टिव हैं कि नहीं है. अगर इंटरेक्टिव है, अगर नहीं है तो यू विल गेट अ प्लॉट इन अ पैरेलल. और बट इफ यू डूइंग विद द, if they are having interaction, then you will get a slope out of that. Then again, randomization, it is very important. Randomization is why it is, if you want to make your study unbiased, biasness, if you want to reduce the biasness of the experimental designing, then you have to go for the randomization. What is randomization? For example, I have to go for a study where I am requiring for a study of a drug, I am requiring 20 people, and for the study of the placebo, I am requiring another 20 people. I'm having a bunch of 40 people. So what is randomization? Maybe because if I am studying for a particular disease, means population what I have selected, the samples what I have selected might be their disease somewhere, some will be diseased and some will be not, some will be healthy humans. So it will be always biased. If for a placebo I'll select all the healthy humans, and for the drug I'll select the diseased samples. So यहाँ जो आपका biasness को reduce करने के लिए always go for a random sampling. Random sampling means कि आपके पास 40 का पूरा bunch है, you are not aware कि that is a blind, and blindly you are selecting the 20 samples for drug study and 20 samples for the your placebo studies. Then what the result you will get, what the result you will get, it will be unbiased. So biasness बहुत कम होगी when you will perform the randomization in the experiment. ठीक है, तो randomization is again very important when we talk about a successful experimental designing. Then comes one more term, it is very important term which is coming, that is called blocking. Blocking, you will every time you will heard a term blocking. So blocking again, this is maybe randomly you are performing the experiment, but at a certain time you are limiting some factors, or you are blocking some factors which are you are thinking it is going to affect your experimentation. For examples, you want to test performance of a fertilizers and you are on a particular crop, but again you are using it on a different type of field, and you are finding that field will the field selected field is going to hamper or going to affect your results. So here I can block this factor. Okay, I'll not consider this factor, or I'll block it. So maybe like that. One more example you are having, okay, for you are going for a formulation of some you can say tablet or a capsule for like that, and you are taking the raw materials from different type of bags, and you were having idea that the raw material what I am taking from the different type of bags, then they can also affect my the performance. So here I can block the factors which is associated with the raw material or bag of the raw material. So this is called blocking. Using the in experimentation you are using the randomization, but you are blocking few parameters which is you are thinking that it is going to, it can vary your results. So this is blocking. Then now comes the type of experimental design, that how we are doing. So if you'll see the experimental design, we are having two type of experimental design. One is screening design and another is response surface design. So in the screening design, here we are using this design in which we are selecting the factors which are most significant. Here what we are doing, we are screening out. Okay, we are having 5 factors or 6 factors, and by these methods we are selecting which will be the very important factors which you want to consider. taking into the what the expected response is there. For example, sometime my target is drug loading on a carrier system. Sometime size is very important for me. Drug loading is not important. Size, primary target is size. Size is there. So there are so many factors which will be influencing such type of result. So when I am planning my result, screening designs are very important to limit or to give you the factors which are most important or most significant factors which are changing, or which are what you can say affecting your responses. And the second design, the experiment designing, is response surface design. In this design, you used to determine the optimal setting for a significant factors. Finally, you have selected the factors, you got the factors, then now you are optimization for going for the optimization of the various factors. You are getting with these factors, you are planning for the optimized formulation. So in this, the response surface design, we are doing it. Okay, so this is the this is one of the figure I have given. If you'll see current setting, what I am presently I was having, and this is, you can consider a hill, or I want to go on the top of that. So currently, sorry, you are having a current setting, which will, when you will go for the screening, after screening, you will get improvement, and you will get some, you will move higher, and you are having a particular situation. In that, if the factors, with the factors, when you will do with the optimization, then finally you will get the peak out of that. So it is systematically improving, and you are going into the domain or the result. You are going towards the achieving your aim in a very pinpointed or very in a accurate manner. So now these are the some various models or experimental design what we used to perform in these two type of methods. For in a screening method, you are having a Plankett-Burman model, experimental design, factorial design, 2-level full factorial design. And in case of optimization, you are having a Box-Blanken design, you are having a central composite system, and you are having a 3-level factorial design. So these are, again, we can discuss every design in detail, but just I'm giving you, as I said, I'm giving you the overview of experimental design. So these are the few experimental design what we used to perform or we can go for. Then comes the optimization of achieved by going through the various, I was talking about the various optimization. So this is the, again, simplified process I have given you. First is screening, then improvement, and the determination of the optimum. That as in the figure also I have represented, and here I am giving you this is the way in by which you can. So in a screening, you determine which factor really influence the outcome. Then in improvement, the various approaches, optimum by the repeated changes or factor settings, or you can say the in a selected parameters, what the changes you are making, and then finally determining the optimum in which you are getting various eigen values you are getting, and you are achieving your aim there. So in brief, if I'll say, these are the various 9 steps of designing of experiment. First, define problem to solve the objective using designing of experiment. क्या करना चाहते हैं, क्या problem select करते हैं, क्या objective है, this is very important. After that, you will go for the determining of the response and their measurement. What the responses you are looking for, how and what the measurement, or what the values you will give it for. Then after that, you will go for the determination of factor and their labels. Factors मैं बात कर रहा हूँ, फॉर एग्जाम्पल जैसे मैंने बार बार आपको बोला, फैक्टर क्या है, स्टरिंग स्पीड, और स्टरिंग स्पीड तो एक फैक्टर हो गया. अब उसके लेवल्स क्या है कि मुझे स्टरिंग स्पीड कितनी रखनी है, 10, 20, 30, 40, 50, 60, 100. So determining about the important factors and their level. Again, this will be done through the experiment, through the designing you will do. Then after that, determining the suitable experiment design. Okay, you got all the parameters, and now you want to decide for the, so then we will decide for the suitable experimental design, which will be most, which will be suit most to the selected data. Then after that, determining the experimental run based on the selected design. How many runs the software is predicting and how many runs we have to perform, that will be, you have to decide. Then conduct experiment and collect the data. There are two type of, once the software or once the experimental, through the experimental design, you have selected the experimental design, you have designed your total number of runs. So you will run actual experiment on the data, and then you will go for the analysis of the data and feed into the design of design of experiment in the software. And then you will interpret the result in the light of the set objectives. Finally, you will verify the result, conduct that, if it is required, you will conduct the additional result if required. So here what we are doing, in short, I am telling you what actually with the experimental designing or software we are doing. First, we are deciding our priorities. Then we are deciding, after deciding the factors, we are setting the limits, or we are giving the design area, we are giving setting, and we are all the parameters we are providing into the software. So what I want to do, which are my various important factors, what will be the factor label I am considering, depending upon the type of experimental design I have selected. The software will predict you value of that. And after that total number of runs what the software has predicted, you will perform your actual result. On the basis of that, you will get the actual data, then you will put into the software. Again it will analyze and give you either what it is predicted you have achieved it or not. If it is going with that, it is going with the fit, then you have the deviation is very less, then you have achieved the maximum and you will get the very good result what you have targeted with this. So this is the whole process of experimentation. This is again, I have kept the checklist when you are going for a design of experimentation. This is the major checklist you should consider. Clearly state the object of experimentation, what exactly you want to know. Check constant and on experiments, constant factor combinations and or changes, constant on size of the experimentation. Again, you have to decide. So this you have to give, so accordingly your software will give you the various runs. Make sure the measurement are obtained under the constant external conditions. If not, apply. अगर ऐसा कुछ नहीं हो रहा, जैसे मैंने आपको पहले बोला कि अगर ये उस तरह नहीं जा रहा है, तो you go for the blocking. Include various center points to validate model assumptions. Check of constant variance. Check of non-linearity, it should be done very. So again, these are the topics on which we can talk much, because this is one part of my presentation where I have given you. After that, the second part is there, but we are having some that I have already crossed my total time which was given to me. So just I'm closing it. So this is first part. So these are the various, you make the checklist. These are few very softwares which are freely available in a trial manner. You can most popular, this is Statis is one of the most popular software for designing, and it is given by the that is Design Expert software, sorry. So this is there, and this is again I have given you the list of various softwares and which are provided your जो is the link I have given you, which are from where you will get it. And to be very frank, it is very now they become very friendly. If you'll go to what I am telling you, if you'll go to the software side, if you'll go for the softwares, if you'll study that, a very systematic tutorials they have given to utilize their softwares. And also they are giving you opportunity to use the trial versions, and most of the trial versions are, they are very effective. The area academic research, what the things we are requiring in the academic research, they are provided with that. So it is now the things are very easy. I'm telling you my story. When I initially, when I came to know about the first time when I came to know about the designing of experiment, I'm talking about in a year 2004-5 when we started working on this. So just I was interacting with one of my friend who was studying in IIT Delhi, and he initially discussed and he told me that there is a something software which is used to predict that. At that time it was a new thing. When I came back, I just searched for that software, and I got that software on the website, and I'm telling you, by trial and error we started working very fluently with that software, just because of with the help of their tutorial, their question and answer. We were writing to them, they were answering us, and we made the process very through. But right now the situation is very changed. We are living in the era of COVID. Everything is available online. If you keep, even I was making my presentation, I have taken support of so many presentation what I have seen in there, where I find that it is effective and I should for understanding I should include it, so I have included. So now the things are more simpler, but using the information in a proper way to get the maximum benefit or maximum achievement, it is with us. So I think so ki I have given you the overall overview of the experimentation and designing. So keep it up. That's all. Keep it up. Thank you so much for giving me opportunity to interact with you. Lastly, I'm acknowledging all my mentors with those who have supported me: Professor V.K. Dikshit, Professor Shailen, Professor Babu Tekwani, who was my mentor at United States, Professor Murthy, Professor Rapka, and Professor Kanwar, who have supported me in all the ways. And right now, still they are supporting me for the various, because I always consider as myself as a learner. मैं अभी भी अपने आप को student मानता हूँ और मैं सीख रहा हूँ, हर बार कुछ सीखता हूँ अभी. तो this is my acknowledgement to my teachers. Also, I am acknowledging various funding agencies who are providing us fund to perform research at our institute. So I am very thankful to them. Thank you so much.
Thank you so much, sir, for sharing such wonderful thoughts with us. So many participants benefited from your lecture. The presentation was very subjective and informative, sir. I want to thank you, sir, for taking time from your busy schedule to join us for this program. Thank you, sir. Now I would like to invite Dr. Anjali Sharma for vote of thanks. First of all, a very good afternoon to all. Myself, Dr. Anjali Sharma, and on behalf of organizing team, Guru Gobind Singh College of Pharmacy, I am very thankful to you, sir. Thank you so much, sir, for sparing your time from the busy schedule and enlightening us with your knowledge. Respected sir, your lecture was really fruitful for the present-day researcher as well as the faculty member, as research has become mandatory for every professional. The session was really informative as we come to know about how our research can be a good research. And for this, selection of research design is must. Based on the design, a researcher can easily frame the objectives of the research. And this lecture on pharmaceutical research designing and approaches in experimentation is really informative for all of us, as the correct research design enables the researcher to proceed in the right direction without any deviation from the task. And at last but not the least, I am very thankful to our participants for patient listening. Thank you all. Thank you once again, sir.
Thank you so much.
Thank you, Dr. Dipendra.
कैसे हैं sir?
Both fantastic, sir. Thank you for your stimulating lecture today. Your presentation is, I think, especially helpful for those doing research and provide much help to them. And you share your years of research experience, your depth of understanding, and your ability to present the topic in such an interesting way. Provide one of the most memorable lectures that I have heard. Thank you very much for giving such a nice lecture, sir. And thank you so much for your time, knowledge, and your presentation, sir.
Thank you, sir. Thank you so much.
Sir, now as a token of respect and appreciation on behalf of Guru Gobind Singh College of Pharmacy, I would present a certificate. It's on the screen.
Thank you so much. It is always very, now we are living in E-era. Thank you so much.
Hey, we are inviting you. Also, sir, you are not coming to our college, no?
नहीं सर, आएंगे, जल्दी आएंगे आपके पास.
चलो इंस्पेक्शन में बताना बस.
आपने जैसे बोला है, अब कोविड खत्म हो रहा है, जरूर मिलेंगे सबको. क्या?
Sure, sir. We will have
okay. Thank you so much.
Thank you.
ये हो गया खत्म।
Participants, now you can leave. We can join again around for third technical session. Thank you to all.
Thank you.